Go, go, go.
Hey, hello everybody. Welcome to yet another edition of our monthly journal club, or sometimes twice a month, sometimes every other month. We'll try to have it every month. Sometimes we have to postpone these things because of life gets in the way, such as faculty meetings. But we are here today discussing a paper on telomeres. So I am Dr. Medvedik, Life Extension Advocacy Foundation, also a professor here at Cooper Union, broadcasting from New York City. And joining us is Steve Hill and Sven from somewhere. Okay, so we're going to be doing another telomere paper, a telomerase paper next time around. So this is going to be back-to-back telomeres. And the paper that we're going to be doing today is Airscreen. There we go. Is the Povedano et al. Paper, very recent, from the Blasco lab. It is therapeutic effects of telomerase in mice with pulmonary fibrosis induced by damage to the lungs and short telomeres. So what's this all about? So this is all, obviously they're studying this because it has human relevance to human disease. And stop this for a moment. And one type of disease that they mention is called idiopathic pulmonary fibrosis. So I had to look up idiopathic, and basically it means that we don't know how it happens. It just pops up. And people, their lungs start to scar. You basically replace lung tissue that's functional, alveolar tissue where you actually have a gas exchange occurring between the blood and the air. And it basically, you have fibrous tissue where you have a lot of extracellular matrix, collagen being deposited, and the lungs become non-functional, lose their elasticity, and basically your lungs stop working. And there are a couple of drugs that supposedly delay or slow down the development. I think they might be antibody-based, they mentioned in the paper, but they don't reverse it, they don't cure the disease, they just kind of slow it down. And the only real cure is something rather drastic, which is a lung transplant, which is pretty drastic. So this paper actually highlights a potential therapy that could work, and it seems to be working in a mouse model that they developed for idiopathic pulmonary fibrosis. And the suggestion here in this paper is that the root cause of this illness that's fatal is short telomeres, or severely shortening of telomeres, and that this can be reversed by lengthening telomeres in situ, in particular cells in the lung. And these cells are, just to remind myself, share screen, are, I believe, AT2 cells? AT2 cells, alveolar type 2 cells, which are a progenitor cell that makes the epithelial cells in your alveoli. So what are they basically doing? Well, in this paper, they use a viral vector, AAV9, and this vector carries the gene telomerase. And I'm not certain here. I have to look into materials methods whether it's integrating or non-integrating. It might be integrating vector, but it is it's expressing telomerase, the catalytic subunit, and this mouse model. So I'm going to just scroll down here where they talk about the mouse model. If I can find it. But basically, we'll probably just scroll all the way down to the actual first figure. Okay. This mouse model is so there's a couple of mouse models. First of all, for idiopathic pulmonary fibrosis, and the early ones are not very good. They don't replicate all the conditions or all the symptoms of the disease. If you just knock out telomerase in mice and you passage them for several generations so they have short telomeres, they do not develop idiopathic pulmonary fibrosis. So what researchers have been doing is using a chemotherapeutic, which is this thing here called blio or bleomycin, which induces double-stranded breaks. And that does induce idiopathic pulmonary fibrosis, or pulmonary fibrosis, since we know what the cause is in this case. It's bleomycin-mediated damage. But once you stop giving the drug, within 2 or 3 weeks, the mice reverse the condition, right? So it's not permanent. So what these authors here found out is by doing kind of a double whammy, and you also have to have a large dosage of bleomycin. But what these authors found is that if you take telomerase negative mice that have been passage for 2 generations, so their telomeres are shrinking generation after generation, and they have now very short telomeres, and you treat them with bleomycin, a lower dose, I think it's about 4-fold lower. I can get the specific concentration is in the paper, 0.5 mg per kilogram, if that interests anybody. Then you get this pulmonary fibrosis, which doesn't go away and eventually be lethal for mice, and it progresses. So it's a progressive condition. So according to the authors, this is a first, of having a mouse model that more closely recapitulates what's happening in humans, for which to test therapeutics. And their therapeutic in this case is going to be the administration of telomerase into the lung tissue of mice using a viral delivery vector and seeing what happens. So and then we'll take a look at the results. So I think is there any questions about that summary?
No, it was very good. I'd just add that they actually designed the mouse model back in 2015 for the reasons you were saying. I believe it was a cancer drug that they used to use, and they would cause insult to the lung tissue using this cancer drug. But as you say, the mice used to recover in about 4 or 5 weeks.
Yeah. And
importantly, it didn't damage the telomeres at all. So this is what's so interesting about this new model that they developed at the CNIO in Spain, in Madrid, is the fact that it does quite closely emulate, the actual disease. As to how close it is, perhaps we can discuss that sort of towards the end. But, I've got some comments about that.
Yeah. Every sort of model, in a sense, is kind of a fudge. You have to, you hope that it recapitulates the most important parts of what you're studying, right? Obviously, it's not going to recapitulate everything, but the most relevant parts, you hope it does. So that's what we have here, and that's what the authors hope, and that's the best we can do without actually strapping people down against their will and testing these therapeutics, which it's ethically frowned upon.
Yeah,
anyway,
it's not the done thing, really. I think we would be a bit unpopular with the FDA if we did that sort of thing, right?
Yeah. Anyway, so back to the paper here. Okay. So the first figure kind of threw me off a little bit because I usually don't see GFP highlighted in red. So I think that's a typo. Sven, I think you caught another typo in the other paper. So what they're doing here is these are basically lung tissue, and this SFTPC is a marker. So these are all fluorescent markers they're using to identify cells. DAPI obviously stains nuclei, so you can see the nuclei of each of these cells. EGFP is, I believe it's on the vector. I just have to, yeah, it's on the vector, and SFTPC is basically a marker for the AT2 cells that are really the most important, the cells, the progenitor cells that are going to be forming the functional alveolar lung tissue. So they zoom in here, and basically what they're getting here is a percentage of, what's the transfection efficiency, right? How many cells are there? So we have, GFP positive cells, out of the total cells. So out of all the total cells, the efficiency is pretty low. It's 3%. So I'm assuming that everything here that's this speckle, all these speckles of green that are, at the perimeter of the cells is on the membrane. And that those are the cells. And then there's, out of all of those total cells that are, I guess, not affected, you have the SFTPC cells. And yeah, I know this green and the red is kind of driving me nuts here. Is 13%. So the GFP transfected cells and SFTP cells are 17%, right? So it's really what they're showing, and GFP, SFT, out of GFP plus. So out of the GFP positive cells that are transfected, most of them happen to be 80%, basically the take-home message, most of them happen to be AT2 cells. So it seems that the vector that they're using is, has they mention, a preference for these cells, which is good. But out of the total cells that are transfected is, rather low. It's like 3%. But as you'll see, this is something that they've mentioned in the paper, that even though it's a very small percentage of the total cells in the lung that are actually, transfected by this vector, they do get a remarkable reversal of this pulmonary fibrosis in this model. So you don't have to aim for 100% or 80% or even 50%. It could be that just a small rescue of progenitor cells may be sufficient to reverse the disease, and that's one exciting point that I think they make in this paper, because that certainly lowers the bar for success, at least at a clinically relevant level. But anyway, here's the protocol. So they basically have these mice. These are these telomerase mice, and then they zap them with bleomycin, give them a dosage. And then after 2 weeks, they either give or not give intravenously this AAV9 vector, right? And then they do a bunch of tests. So this is week 0, and then they do spirometry, which is basically, huffing and puffing. For humans, it's huffing and puffing into a tube and measures your lung capacity and all your other statistics that your lung gives. I don't know how they do it for a mouse. They probably stick a mouse in a chamber and measure the volume that way. Sure, there's no little tiny mask they put over a mouse's snout. And then they do histopathology, which you see all of these cross sections, and biochemical analysis to see what's actually going on after they treat or not treat the mice. And they have these little tiny MRI scans of mice down here, and AAV9 empty is obviously your negative control. AAV9 tert is your telomerase expressing AAV9. And this is, I guess, this is a cross-section, a transverse traverse section of a mouse. I didn't go to medical school. Versus a longitudinal. So these, I think, are lungs on either side. It's probably the spine. These look like little ribs here. And this area that's light, I guess the dark area is a good lung, and this area that's lighter is more fibrous tissue. So after they stop this, don't add bleomycin, you get some recovery, but, they measure the area here. And you could see from, this one representative mouse sample, and they have several. So here they go, full change volume. And D is N equals 9, so 9 mice. They can measure the volume or the area, this cross-section, of the fibrous tissue, and see if it stays the same, increases, decreases, and lo and behold, it's decreasing over time. And this is just basically, a more quantifiable graph that these little asterisks mean that it's statistically relevant, that you have a decrease down to almost 0, right? So after, so this volume change kind of starts to plateau out for your empty negative control, but it, maybe it'll eventually go down. I'm not sure in this model, but this certainly, bodes well for the efficacy of the treatment. And then what they do here, so in this these graphs, this is basically now, the physiology. They do this pulmonary function, which is, I looked up lung resistance versus I forgot what the C dyne is, but basically it's the elasticity of the lung tissue, right? So basically, so the higher the number, so the more LR you have, the more resistant the lungs are to stretching. So if there's fibrous tissue, it's not elastic. It's not going to be stretchy. It's going to be kind of tough and hard to breathe. And so if you have much higher resistance versus, you know, elasticity being smaller and smaller, then you have a much larger ratio here, right? So the higher the number, the worse off these lungs are. And they probably measure this in, you know, using their spirometry data somehow. So here we have negative controls without bleomycin. These are the numbers of mice. Then bleomycin AAV9 empty. So this is your, in this case, your positive control, which is going to be pretty bad condition. And then you have your treated mice, right? And this is weeks post-viral treatment. And you immediately see just by staring at this graph that the black, which is the treatment, goes down pretty close to wild type, or not, well, not really wild type, but without bleomycin. So, you know, I'm not going to delve into this further, but you can, it's pretty obvious here that there's a difference, and it's quite dramatic. And then, of course, they quantify TERT mRNA expression levels. You know, is it actually because the telomerase is being expressed, or has it got something to do with something else? But yes, the telomerase is being expressed. So I think they do a PCR, probably a reverse trend RT-PCR on this. So anyway, okay, G. So this is interesting. G is, just to scroll down, so histopathological analysis. Where's that G? It uses Masson's trichrome staining from lung sections. I believe Dr. Masson must have been around probably early 20th century, late 19th century, where all of these histopathological stains were developed. And what Masson's stain here is doing is, I believe the bluish is supposed to be areas that have collagen, so fibrous tissue. And I actually looked up the protocol for Masson's trichrome staining, and it looks like a pain. We're not talking about throwing antibody on it. I mean, yeah, antibodies are also hard to work with when you're trying to stain, but man, this thing is like, it's like basically cooking a 3-course meal. Stain these cells and fix them and add alcohol and heat them and treat them, and, you know. So it makes me wonder how they actually developed all of these staining procedures in the first place, since there are just so many variables and steps. But anyway, I digress. So AAV9 empty, you can see more fibrous tissue. And I believe this is after a certain amount of weeks, which probably somewhere in the materials and methods here. What the end point is, 8 weeks post-viral treatment. Yeah, there it is. Collagen fibers in blue, nuclei and erythrocytes in red. And here's affected mice. They have percentage. They say, I don't know how many of these mice, assuming they say here the number of mice analyzed per group is indicated, but it gives a percentage, and I don't see it indicated. So I'm going to assume it's probably on the order of the numbers we see here, which is either 9 or 4 or 6 or 5. I don't know. But anyway, percentage affected mice after 8 weeks, AAV9 empty, they have a scale: severe, mild, non-fibrotics. Non-fibrotic obviously is, they can't see any fibrotic tissue according to criteria they use. I think severe is 30% or more fibrotic tissue, and mild is something less. So in the AAV9 TERT, half of them have non-fibrotic tissue, and the other half mild, and all severe for the other mice. So using, you know, these metrics, so pretty impressive, you know, pretty neat. You know, that being said, it's a model that they developed here. How relevant it is to humans is yet to be seen. But, you know, so far, I think this is the only paper or first paper where there's something that can potentially be a cure, and for a harmful disease. So I'm going to stop sharing this and show my face, because I've been told people want to see my face occasionally, I guess. I mean, there's a reason why I shave every day, right? Okay. I'm just taking a breather here while I scroll to the second figure. So I don't know, any questions about what we just looked at, saw? No?
Seems pretty clear. I mean, it is pretty straightforward, the paper.
Mm-hmm.
Generally speaking, I've seen much more confusingly laid out papers. It's pretty straightforward, and the results are quite significant. I mean, looking at it's very obvious, which is, well, I was personally really impressed by it when I saw it last week. I was like, oh, good grief. So so the big question really is, how relevant is it to actual true fibrosis? That's the big question.
Well, I think even a deeper scientific question for me is looking at the connections between damage that's distal to telomeres, right, and the telomeres themselves, right, because the two obviously go together. There is a connection there. They probably share certain repair factors, and so the fact that longer telomeres could help mediate damage occurring elsewhere, I think that's a very interesting connection that needs to be, further fleshed out as far as at the molecular level. They don't really go into that at all in this paper. They just mention that that's what happens and that's that. But at the molecular level, I would be interested to maybe pursue that a little further at this just a purely scientific level.
Well, I mean, Blasco showed, I can't remember what year it was, 2012, 13, something like that. They demonstrated that telomerase does increase stem cell mobility, and there's been quite a few other papers sort of suggesting, well, showing it really. So, and I recall that they did an article in the news about this particular study, and they were saying essentially it leads to a better repair program, is how they explained it, rather than a poorer or secondary or backup repair, which is fibrotic tissue. Now, and I know that another study by another group showed that if there's no healthy tissue available, like the cells that form the tissue, then scarring or fibrotic tissue tends to be the result. The fibroblasts jump in and make a poor secondary repair. So best guess what's going on here is the telomerase is improving stem cell mobility so that the stem cells are more active and they're producing more available healthy cells to replace, like the lung tissue. So then the fibrosis isn't happening because your fibroblasts are not jumping in to create a poor secondary or backup repair. That's based on other papers that are not related to that, but it seems to suggest that might be the case.
Sort of like the cellular version of NHEJ versus homologous recombination.
I guess.
Boy, that's such an esoteric analogy. I don't even know if that qualifies as an analogy.
But yeah, they that would be the best guess. That it improves the mobility, and that kind of ties in again with what the Conboys have done as well and demonstrated. They went a different route and inhibited TGF beta to improve stem cell mobility. But what they found is when they improved the mobility of stem cells, then the tissue regeneration was much greater and better. So it all sort of ties in for me.
All right. Back to the paper and see what else the authors find. Okay, on to figure 2. We have lots of bars here on this figure. Look at all those bars. And this is actually the result of, so before I jump into this. They look at cytokine levels that are markers of inflammation, right? So these are inflammatory markers. And it turns out that a company, I forgot which one, it makes a kit which is conveniently has a whole slew of these markers. So they do ELISA on this. And when I first heard about it, I was like, oh my God, somebody had to do like 96 well plates, like, because that's how I used to do it back in the 1990s as an undergraduate. But no, it's like a little chip you get that's like an ELISA, I guess, on a chip, which you just basically see everything at once, all in parallel, which is pretty cool. Anyway, so AAV9 TERT treatment leads to lower collagen deposition, which we saw earlier, but they use other markers, right, besides this Masson trichrome staining, less inflammation, and decreased active fibrotic foci. So they're just kind of going a little bit further here to show you that, you know, that looking at all these other markers that indicate there's less fibrosis, and there actually is less fibrosis. So they use this Sirius red staining, polarizing light. So this stain basically, you know, if it's bright orange, which kind of looks yellow for me here, but it's supposed to be indicative of, I believe, collagen. So that's more fibrous tissue. So this is AAV9 TERT. I think this is post 8 weeks, and so you could see a difference here. And this is collagen deposit in a certain visual field percentage, which, I don't know, it goes down here, obviously, but I don't know, it looks like a much bigger difference to me here, visual field, than here, so I'm not sure exactly what cross-section or how they're measuring this. But anyway, the point is that there is a drop, right? So, they also look at hydroxyproline collagen peptides, which hydroxyproline is a component of collagen. It's a modified proline. And they look at these peptides that are also indicative of more collagen. So without bleomycin, it goes up, boom. And then full change of this normalized to AAV9 empty, when you use AAV9 TERT, it goes down. So that's kind of fitting with what we've seen so far. Normalized pro-collagen levels. So again, normalized, I think, to AAV9 empty. Normalizing just means you compare, you set your, whatever your control is, you know, you set whatever you're comparing to the control, and then becomes, the control becomes 1, right? So pro-collagen is the precursor to collagen. So again, 3 weeks post-treatment, 8 weeks post-treatment, they give a percentage here decrease. I'm not sure why they stick a percentage here when they're somewhere else, but anyway. AAV9 TERT, it goes down, right? What else do they look at here? This is a Western blot. Again, you're comparing 2 billion, which is a control, to TERT levels. You can't really see this here. They do a quantification, you know, to show that pro-collagen levels go down with TERT expression. But, you know, I mean, this is darker than that's darker. So you really got to, you know, measure that with the computer to you know, get a ratio. But anyway, all of these numbers basically show you that that yeah, this is dropping however you look. And then they look at H, which is so it's macrophage specific markers. So macrophage is obviously, you know, if it's some inflammation happening, you have more macrophage infiltration. And they have this marker called F4/80. It's the weirdest name for a protein. I don't know what it's supposed to. I just did a cursory glance. I'm like, why do they call it F4/80? Some kind of ratio, but it's just a marker. And this is found on macrophages and augmented. I'm not sure what augmented means here. I mean, they're zooming into this, but you could see that, you know, MTVEC, you see this macrophage specific marker that's, so there's a higher percentage of cells in the view. So empty, TERT treated, right? So this is another marker for fibrosis. And then another one, yet again, are these myofibroblasts, which I looked up what are myofibroblasts, and they are something in between a myoblast and a fibroblast. There you go. So, so it's something that's somehow turning into a fibroblast, I guess, a myoblast. So this is also indicative of fibrotic tissue. So this alpha SMA marker, so it's present to a higher degree than in the AAV9 TERT after 8 weeks post-treatment. Okay, so they're checking all the different check boxes that you would have for fibrotic tissue. And of course cytokines that are indicative of inflammation. So 3 weeks post-treatment, 8 weeks post-treatment. I'm just going to skip to the 8 weeks post-treatment. Redundancy. And you could see just by glancing at this that AAV9 TERT, levels go down, right? So significantly all across. You get this weird jump in TNF alpha and what is that? TREM1, I think. Temporarily, I'm not sure why. I could dig deeper and see why, but anyway. I don't know what this means. No, it's not significant. This looks significant to me. But anyway, looking at this entire panel of things that are pro-inflammatory, it's pretty obvious that they go down, right, with treatment. Okay, so I'm going to stop sharing here. Cool. All right. So they're looking at a lot. They're looking at everything that they can think of to show that this fibrotic tissue is going down, and all the things that are consequential to fibrotic tissue, right, that come as a consequence, also go down. Everything you would expect, that fibrotic tissue manifests as a negative. So that's figure 2 in a nutshell. Yeah.
It's impressive.
Yeah.
And I got to say, it's very, very thorough.
Mm-hmm.
I mean, the huge range of things that they tested for was, well, impressive.
Yeah.
I mean, some of your favourites are in there, the usual suspects, and it's interesting, as you say, to see that they're all reduced.
Yeah. So that's a good sign. Again, the reason they're going into this depth too, which is good, is that there's really 2 things that this paper is really kind of opening up. One is a potential therapy. One is this new model that they're developing that nobody has developed before. So you have to convince people that that hey, this is a legit idiopathic pulmonary fibrosis model, right? So your hope is that other people adapt this model for their studies. So you really have to take the extra mile and characterize it. And I'm sure reviewers asked for that too. But I'm sure they knew that as well, right? Because it's there's really 2 new things, 2 new sets of ground that they're breaking here. And if the model is bad, then their therapeutic is already shaky, right? And and if their therapeutic is shaky, well, they need to show that it's shaky because the model isn't shaky. So they've got 2 things to really kind of cement here, which they're doing a pretty good job. All right. Move on. Share screen. Yet more markers. All right. Figure 3. AAV9 TERT treatment reduces DNA damage, improves telomere maintenance and proliferation in AT2 cells, right? So that's what you want. You don't only want to have less fibrotic tissue, but you want to have more of these AT2 cells, and as a consequence that they talk about in this paper, that it is as a result of telomere maintenance, and also that if you add AAV9 TERT as a consequence, you reduce DNA damage. And here are all your usual suspects for DNA damage. AAV9 empty, AAV9 TERT. This is histopathological sections. They try and look up the antibodies they're using for staining. But you can see lots of little red arrows here, lots of little red arrows here, and this is stains for various indicators, so gamma H2AX. So this is a histone that's phosphorylated in response to DNA breaks and damage and recruits repair factors P53. The big one here that's involved in basically inducing apoptosis or repair, et cetera, they're kind of the master regulator for, cells avoiding cancer. P21, tumor suppressor. C3A, is that a C3A? C3A. What was C3A again? Embarrassed to say, it just slipped my mind. Anyway.
Isn't that a complement? C3?
It could be, but I don't think it's that. Caspase 3, caspase. Yeah.
Oh, yes.
So caspase, obviously. So it's an indicator of apoptosis. So obviously, if damage is occurring, more cells are being apoptotic. So all of this stuff goes up higher. Then they quantify it here. So these are cells in the visual field. So that's what all these bars are. So you get drops. So I'm not going to really, And of course, SA beta gal is beta galactosidase, which is our old favorite marker for senescent cells. So more blue speckles here than there. And you can see these numbers recapitulated in this bar graph. Okay. And then, of course, they look at gamma H2AX, SFTPC positive cells relative to SFTP cells, SFTPC cells. I don't know why they just call them AT2 cells, because that's what this is a marker for. But anyway, it's kind of driving me nuts switching between the two terms. But you can see less. So the cells that are labeled with SFTPC, which is brown, versus gamma H2AX, which is red, whatever fluorophore whatever it's conjugated to that recognizes that, they're less, right? So less of the cells that are going to be the AT2 cells that are the cells they're concerned about, that are the necessary cells to induce repair or exhibit this repair marker. So not just cells in general, but they look at this one repair marker, they decide to apply it to AT2 cells. So, what else do they do here? Got to remind myself, because now they're looking at telomere levels. Go down to the figure here. Fold change in telomere link. So let's see, is this, so they say this is significant. I didn't look to see how they did the telomere length measurement. I think they mentioned in the paper they actually, I'm not sure, this is the one thing which I'm not sure how easy it is to quantify, because they use fluorescence intensity and a marker. So they use fluorescence in situ hybridization, I believe. I could be mistaken, but to coat the telomeres, and whatever the telomeres bind to, whatever it binds to, you're going to have speckles wherever it binds to the telomeres, and the more binding you have, the more brighter these little dots are, these foci, and then you can quantify that. I'm not, so that's the measurement they use for telomere length. So they show it goes up. I mean, this number is, this is relative to non-AT2 cells. So maybe I think it's significant. Is it non-significant or significant? I'm not sure, but it might be significant. It's kind of a little bit. Percent AT2 short telomeres relative to non-AT2 cells. So empty TERT. So the percentage of cells that are AT2 positive that have short telomeres relative to non-AT2 cells goes down. And here is their fluorescence data. Let me see if I could zoom in on this. Is that bigger? Let me just open this up. Can we see this? Okay. So, Psi3 is a fluorophore, and it's used to label telomeres. It's conjugated to something. Either I'd like to say it's a small piece of DNA that's part of the telomere that it's hybridizing to, but it could be a telomere-specific protein. I have to double check that. This is your stain for SFTPC. So this is 10 microns. So these are cells, and this is your DAPI here, so pretty big nuclei. So these are your cells that they're concerned about. I guess a little less speckling, but anything that's got these speckles is supposedly an AT2 cell. And all of these and DAPI and all these little red speckles are supposed to be telomeres or parts of telomeres. And basically they zoom in here, and what they're looking at is intensity of these speckles. So you can see what I'm saying. It's a little, you know, I mean, you have to, obviously you can't do this by eye, you have to do this computationally, right? So, and they're saying that if you look at, I think, the brightness versus the area, you get more here than in here. That's the comparison, right? So that's their measurement, and that's how they're measuring it. So, you know, there might be some issues with that, but whatever. I mean, they tell you exactly what their measurement is, and that's it, right? So you could probably, you know, I don't know, I didn't look in the supplemental. There are other ways you can measure telomere lengths, obviously. You know, you could run them on gels. You could do various, you could do other assays that, you know, directly measure the lengths of nucleotides. So I'm not sure if they followed up with that, or this was their main way of measuring. Okay, so let me zoom back out, get this out of my face. Okay. Too big. Let me get this smaller. All right. Okay. So now they look at cells that are proliferating. So they use this marker Ki67, which is a marker for proliferating cells. Forgot what it exactly marks, other than proliferating cells at the molecular level. So when you have TERT, more of these cells in the field of view are proliferating. Again, here's the representative data on your right. Is DAPI stain, SFTPC or AT2 cells, and then Ki67. So you could see that you got a lot more red splotches all over here, and that I believe some of these red splotches correlate with the AT2 positive, or AT2 cells, sorry, alveolar type 2 cells. So, and these are the bars that basically tell you. So here are your SFTPC positive cells. You got more of them, you have more proliferating cells in total, more of them are SFTP cells, and out of these proliferating cells relative to SFTP cells, more of the AT2 cells are proliferating. So that's kind of the more important thing here, that out of all of these cells with proliferating marker, the marker for proliferation, a lot of them, a lot more versus AAV9 empty is, you know, is proliferating. So not only you have more of these AT2 cells, but they are active, right? They're dividing. Yeah, so I'm going to stop sharing this for a minute. So while I scroll to the next figure.
So that staining would perhaps suggest that there is definitely improved stem cell mobility.
Yeah.
The stem cells supplying the tissue with, you know, the actual cells that they need, like I was saying earlier, rather than the fibroblasts doing a bodge job.
Yeah, it's interesting, you know, how we define stem cells, because these are AT2 cells, they're progenitor cells, and there's most likely progenitor cells to those progenitor cells, right? So it's like, be interesting to know which cells are capable of upregulating normally telomerase, and, you know, when they upregulate. I mean, it's still, stem cells is still a messy field, right? It's going to be messy for a while until we really know how to distinguish which cell is. I mean, people are distinguishing them, but there's a lot of fuzzy edges to all of these definitions.
Yeah, I mean, it's a bit of a fudge, isn't it? I mean, there are different types. There's like, I was reading a paper where you've got stem cells and reserve stem cells. This was actually in the intestine.
Mm-hmm.
And again, this was to do with telomeres. And, well, we're going to talk about this next time, hopefully. But they found every time it makes a repeat, to top up the telomeres, every time there's a GTG or whatever it is, I can't remember what the sequence is, it keeps doing that. But in that little program, there's a pause button, almost like a pause function. And because that's there, it slows down how quickly it can top up the repeats. So it's, it's really interesting. And it sort of ties in because when they remove that pause, the stem cells became super efficient and they could replace the repeats as fast as they would have, being removed. So that's interesting, and it all sort of ties in with this.
Mm-hmm. So yeah, I mean, again, we could talk about this later, but maybe next paper, but there's always been that fear that, I mean, even in the hallmarks of cancer paper, right? Mm. Telomerase upregulation is one of them, right? Cancer cells have to be able to fix their tumors.
Yeah, it's a step. A lot of people think that it may induce or encourage cancer.
Yeah,
but it's not. It's not really that simple.
Yeah, it's a lot of other steps.
It is. It's not that telomerase causes cancer. It's just that a cancer cell needs telomerase activity or ALT. Sometimes, very rarely, it uses ALT, which is alternative lengthening and telomeres, to basically immortalize itself and proliferate with that end. But it's not that telomerase actually causes the cell to go, I'll be a cancer cell.
No.
It's far more complex than that.
Yeah, it's basically there's like 7 things categories of mutations you need to have to get a full-blown malignant cancer. Maintenance of telomeres is one. So if you have the unlucky cell that has 6 of these mutations, then that telomerase is going to push it over the edge, right? But otherwise, it's fine.
But yeah, the gambit here is it's essentially, can we walk the line between, on one hand, extremely robust regeneration, which is seen in species like salamanders, Mexican salamanders in particular, and even us as humans in the first couple of weeks of development have this ability to regenerate fiercely. Starfish also have that. So can we, on one hand, can we have that? And on the other hand, you've got cancer. Can we sort of walk the line in between and balance it?
Right. Or is there a mechanism in place during stages like development and species that enables telomerase to be on all the time, active, and have some sort of alternative extra suppression step happening, that ensures that you don't have cancer, or can we learn from that? Can that be a trick that can be employed here, right?
Yeah, it's a bit like having your cake and eating it, and certainly companies like AgeX appear to be sort of working in that direction. They're experimenting with telomerase activation, and it's coupled with what they call ITR, which is like some kind of it sounds like cellular reprogramming, like OSKM. So they seem to be wanting to reverse epigenetic aging and telomere attrition at the same time. So they seem to think that we can probably walk that line between the risk of cancer and having the regeneration that we want.
And we won't find out unless we try.
We won't find out unless we try. And fibrosis is a great start because, there's no effective cure.
Yeah, and who wants to have a lung transplant?
I don't fancy that.
It's pretty rare, and so I'll take a telomerase therapy first. See how that goes. That's just me. Anyway, back to the screen. So here, it's really not so much. So figure 4 is basically they do a lot of microarray studies here, and I'm not going to go too much into this. They do a statistical analysis to show that this is all. So this is going to be a fast figure because we don't have the raw data here. But basically, they're looking at and here they just box it. It's probably somewhere in the supplemental, but they're looking at what classes of genes go up when you have this therapy, and is it all just like with your fibrotic tissue, all your markers of fibrosis that are inflammatory go up. What goes up when you add AAV9 TERT, right? So what are the transcriptional changes in the lungs? And what pathways are upregulated A and downregulated B? So, what do we have here? Somebody very soon upregulated and downregulated by AAV and TERT compared to AAV empty, 8 weeks post-viral treatment. And let's see. So clearly these go up. Apoptosis, I don't know, is that supposed to go down? Or is there like a reason why it's orange? I'm not really sure. They do mention it goes down. DNA repair, let's see. Well, they say this goes up, so that's fibroblast activation. So that's going to be pathways that are going to be required for these cells to become, fibroblasts, and then thereby lay down fibrotic tissue. So that goes up. Make sure I get. That goes down with TERT treatment, and this goes up. So here they have apoptosis. I don't know. They certainly had less apoptotic cells, so I'm not sure why the markers for apoptosis go up. Certainly, it makes sense. DNA replication and mitosis goes up because you have cells replicating. So obviously, everything that's going to be involved with that is going to go up. DNA repair, right? So clearly, if they're implicating telomeres, have something to do with that process. Markers are going to go up in that, and others. Extension of telomeres, that goes up as well. But apoptosis, not sure why that would go up. Apoptotic execution phase, cleavage of cellular proteins, apoptosis. So, again, this is just a box here. I'd have to take a look at see what specifically is going up. This is just a summary, really, and statistical analysis showing that these things are relevant, that the levels of TERT, extension of telomeres goes up, for example. And these are pathways that are involved in, fibrosis. So canonical Wnt pathway, this is a signaling pathway. Full change in messenger RNA, let me show that it goes down. So this is like really some of this data here that's, I guess, shown here as a bar graph. And BID, BIM, FLIP, apoptosis. Are these pro-apoptotic? I keep forgetting. BID, BIM, FLIP. Not sure. I would say that, maybe it's apoptosis of fibrotic tissue, but then again, they had less cells that were, let me scroll up here. Right, so there's more, I don't see any apoptotic cells here, right? At least this marker for caspase. But anyway, I'd have to dig a little deeper to see what's going on there. So that's figure 4. And I think there's another figure here. Whoops. Figure 5. So here they use FACS sorting, so fluorescence activated cell sorting, to isolate AT2 cells and to show that they're overexpressing TERT and they're downregulating damage, TDR and inflammatory related pathways in these particular cells. So this is just scatter, the types of cells. So they're picking cells that are So CD31, CD45 negative are, I believe, epithelial. So that's the class of cells the AT2 cells are. There's a marker for these cells. So this is the population that they're isolating. So here it just shows that they've isolated these cells. That's the SFTPC messenger RNA, and the TERT levels go up. Obviously are higher in these cells that are expressing. P53 messenger RNA levels in those cells, the AT2 cells, are down, and the P21, so tumor suppressor, P53 master regulator, tumor suppressor, go down, right, in TERT. And this is just statistical analyses that I won't go into that show that this is relevant. I forgot what this stands for, FDR, false determination something, number that correlates with how relevant this is, right? So far, you know, you know, they do show TERT levels go up. You know, you're expressing it, the vector's working, all these markers go down. As far as lengthening of telomeres, I want to read this a little further later because they do just that one assay to show that the telomeres lengthen. You know, is TERT overexpression causing all of this primarily due to telomere lengthening, or is TERT doing something else as well, right? So regardless of one or the other, it is working, right? But, you know, I think I'd like to tease that apart a little more to see, you know, and this is more from a scientific perspective rather than a clinical perspective. What are all the functions of TERT in this case? Is it just, you know, it suggests that it's the telomeres, but it could be additional stuff that TERT is doing. I'm not sure how you would tease that apart. Maybe you could make a catalytically inactive TERT that somehow has other functions and see if that also works. Anyway, just thinking out loud here. Scroll. We did that as well. And here we come to a summary of the entire paper in picture form, which makes things easier. Where's the button here? Can't seem to find my, oh, here it is. So, bleomycin and telomere shortening leads to injury of alveolar epithelial cells, immune response, fibrosis, bam. It's kind of cute how simplistic this picture is compared to all the other detailed stuff they have there. Wham! It's like.
But then Blasco's quite renowned for doing things like this. I mean, to use an example, I'm going to mention hallmarks of aging.
Yeah.
But it's a tremendously complicated paper, as we discussed in a previous journal club. But it is accompanied by these really handy illustrations that sort of say, hey, look, I can sum it all up in one page. And you're like,
yeah.
Wow.
And using Microsoft Paint version 1.
But, you know, it's great. It helps. But, it really does help. And it means that everyone can sort of engage with science, which is great, you know.
Oh, I'm not saying that, well, maybe slightly facetiously. I'm a big fan of Microsoft Paint. I like the kind of, I mean, you know, it's kind of back of the table napkin sketch. I mean, look, you've said everything you need to say. You did all your, you know, it's like you've got all the most important data. And if you want a nice, pretty picture, fine. Let me quickly sketch it out for you. There it is, right? I'm not gonna spend a month with Adobe Photoshop making, you know, all sorts of cells that look like they're, I don't know, coming out of a, you know, sci-fi movie or something.
It gets the job done, and it sort of conveys what's going on, which is, you know, really important, you know, in an infographic or you know, that sort of thing. I mean, you're talking about the man here who someone said, what's epigenetic alterations? And I got a colander. Yeah, I got a colander, and I demonstrated with my hands, and I said, imagine my hands are methylation, and the holes are genes. And I said, when you're young, you start out like this, but as you age, my hands start to move, and different holes are uncovered. And they're like, oh, I totally get it.
This is the first time I heard that analogy. I think that's a nice analogy. That's pretty cool, and it's physical. You know, that would be nice for kids to demonstrate, or I mean, not just kids, but anybody. But that's a neat tech. I'm going to steal that.
It was my wife, and we happened to be in the kitchen, and I saw the colander, and I went, aha. I said, I'll explain what epigenetics is in literally like 30 seconds. Cool. It worked.
Excellent. Yet another reason to take up cooking, which I enjoy doing as well. All right, so that summarizes the paper we just read. I'm just going to go back to the title again. Let me just scroll through. See, I'm getting better at switching back and forth, so that way you don't have to look at me scrolling and kind of go into a seizure. So this is the paper again, Pogadano et al. If I'm pronouncing that right. Therapeutic effects of telomerase in mice with pulmonary fibrosis induced by damage to the lungs and short telomeres. Good title too. Boom. Just it kind of explains it. No acronyms. I like that. I think I read a CRISPR paper where CRISPR, the acronym, was part of another acronym. And I just wanted to throw up, but so none of that here. And this paper is open, so anybody can go online and get it. So so don't email Steve and say, can you send me the PDF? It's on eLife.
Well, you can, but I'll just say, here's the link.
Let me Google that for you.
It's already in the actually the paper. I took the liberty of linking it in the description of the video, and I'm going to say again in Dr. Blasco's favour, a lot of her papers tend to be open access. A lot of them,
yeah.
Maybe the odd one or two not, but the majority of the papers that are coming out of CNIO and especially from Maria Blasco always tend to be open access, which is great. You know,
yeah, good for her. I mean, that's the way it should be. And that's the way science is slowly and fits and starts moving towards.
Well, now we are talking about CRISPR. Did you notice there is a new journal on CRISPR by Marie-Anne Libert publishing?
So a journal just on one type of
just on, I think it's called the CRISPR Journal. Actually, yes. And their articles have really funny titles. One of them, I was just reading today the article, and the title is Genome Editing BC, and then between brackets, before CRISPR, Lasting Lessons from the Old Testament.
It's clever. But, you know, they'll be talking about zinc fingers and talons. Yes. I mean, some people still use talons.
Actually. They go back to mechanucleases, to the old technology from the past.
You know, millennia from now, when everything here collapses and only those papers are found and the CRISPR journal is found, new religions will be formed. There'll be CRISPR religion, the talon religion, the zinc finger endonuclease religion. Many wars will be fought. Many lives will be lost.
The talons, it sounds like something out of a science fiction program.
We laugh now, but our future generations.
Well, they will spend then a lot of more time than we did at deciphering it. You know, like we spend a lot of time trying to decipher the hieroglyphs from Egypt, but they will spend a lot of more time if, you know, all of our scientific knowledge disappeared and they just happen to find the paper we just discussed today and they see all these images that make no sense.
and just to add a fly in the ointment, I was reading not so long ago, there is a another version of gene editing now that possibly might usurp CRISPR. I can't recall the name of it, but apparently, yeah, sorry, Oliver, you're out of a job, mate. But no, apparently there is a new type of gene editing technique that might be a contender for the crown.
Do you remember any snippet of what this may potentially possibly be?
Nah, I cannot remember, but I will send you the.
You can't remember something so earth-shaking as this?
Yeah, but I hear things like that all the time. I mean, it's like there was a lot of concerns over CRISPR's accuracy, which were quite frankly debunked, weren't they? But more recently, I heard that it's not that great for, well, human use, but there are other forms of CRISPR, though, aren't they? It's not all Cas9. You've got, is it CF10? And
yeah. Well, there's certainly papers that just came out that, you know, kind of usurped a lot of the research we've been doing here in the lab, but showing that, you know, if you combine CRISPR with localized increases in localized concentrations of factors that are required for recombination, such as REC-A, you get greater efficiency of gene targeting, which could be applicable not just to CRISPR, obviously, but to any reprogrammable endonuclease. But I don't think that's what you're referring to. You're probably referring to something completely new rather than a combination of CRISPR and something.
No, this is something new. I will scan my files, but there was definitely something new. Another type of technique, you know, separate like Talon's is separate as zinc fingers. There's another technique in the pipeline.
It may be new, but is it better?
Well, that's the question, isn't it? You know, things are changing so quickly. But and just to flick back about the apoptosis measurements they took.
Yeah.
We're being told here in the chat that BID is BH3, which is an interacting domain death agonist, and BIM is BCL2 like 11. BCL2, I know this through Senolytics. BCL2, the superfamily, you got BCL, BCL2, BCLW, BCLXL. Those are the three that are significant in Senolytics, and it essentially, the more it's expressed, the more resistant to apoptosis the cell is. So what Senolytics does is it pushes that superfamily down, and the cells become more vulnerable to apoptosis. And a lot of the drugs that do that are cancer drugs, because obviously they're prepping the tumor to destroy itself.
But BID is an agonist of apoptosis, right?
Apparently so. And FLIP, according to Google, FLIP is the master regulator of apoptosis. Yeah. So high is good. In this case,
apoptosis. But in this case, apoptosis is shown going down. Well, the factors that promote it go down. Messenger RNA levels, what they measure. But when they look at the staining of cells that are actually going through apoptosis, it appears those go down, which I'm not really getting.
No, you want FLIP high, really, because you don't want cells dying.
Yeah, but I'm talking about the figures we looked at, right? So we have pro-apoptotic factors go up with TERT treatment, whereas the amount of cells in the field of view that look like they're actually apoptosing goes down. So,
but then in rejuvenating the tissues, it may be recycling aged and knackered cells. I mean, that might also explain it. Anyway,
overall, I thought it was a pretty solid paper. I thought, I mean, it's a radically new type of therapeutic. It's a completely new model for studying this, this dread disease. And, there ought to be more papers like this. And, I think we'll see next time when we do our journal club what the other latest news in the telomerase field is. And, we'll cover some more telomere papers. I don't know, did we cover, no. I mean, obviously we've been referencing telomeres and telomeres with hallmarks of aging, but is this the first paper that we did that looks at telomerase in a disease model specifically focused on telomerase? In a disease model, we looked at, what was it we looked at?
We've done so many now. We've definitely looked at some of the other stuff. I'll tell you what we looked at. We looked at OSKM, epigenetic reversal.
Mm-hmm.
If you remember, we had a look at Ocampo, Belmont et al.'s paper about OSKM,
mm-hmm.
resetting the epigenome,
mm-hmm.
and we then looked the next month. We followed up with the Blasco paper where she did the same,
mm-hmm.
But was looking at with a view to the sort of telomere side
mm-hmm.
of it. And then of course hallmarks we talked about.
Right, but the telomeres were sort of at the periphery there. I mean, there was a consequence, but this one was like they're specifically adding in.
Yeah, this is probably the first time we've talked about a specific telomerase paper. Where telomeres are the star.
And the next one that we're going to be doing is going to be even more specific to a mechanism that's built into telomerase. So.
Yeah, a special little thing built into the program that TERT uses, and I think it's pretty significant.
And that they suggest, I think, I just quickly scanned through that it might be amenable to drug targeting. Correct?
Yeah. And when they actually did knock it down, the pause switch, they found it dramatically improved the efficiency of TERT creating these DNA repeats.
Yeah, and I find that exciting because this is, again, people come up with these compounds where they say, well, this activates telomerase, and I'm like, well, how does it activate telomerase, right? How does this compound, does it upregulate telomerase? How specific is this? So if this is an actual, mechanism and there's an actual binding site and telomerase is already there but doesn't work as fast as it can, then this is a pretty big deal as far as a mechanistic, potentially an intervenable mechanism that we could target with something that's not as complex as gene therapy, for example.
Exactly, that's what I was thinking. Yeah, it's not as complex as adding a copy of the gene. So, in other words, to amplify the expression of telomerase, you're not adding an extra gene, and you're not doing anything like that. All you're doing is you're taking the innate system that's already there, but you're just making it a little bit more efficient to the point that it actually tops up just as fast or even faster than cell division can take it away. That means your stem cells keep on trucking, you keep supplying your tissues with healthy cells, daughter cells, and win-win. But
more efficient, or we could also say maybe more sloppy, right? Because it kind of loses that ability to stop and check things, I guess. Not exactly sure what that pause function is there for.
Well, they had a good look at it, so yeah, we will
review it.
We'll grill it. We'll grill them, and we'll give it the usual leaf treatment. We usually do, don't we?
Yeah, I already thought of some weird analogy that involved being hit with leaves in a Turkish bath, but we'll talk about that. Anyway, enough analogies. Colanders and eucalyptus leaves. I leave you with that message. Okay. Thanks, everybody. Stay tuned for next time. And next time is coming up soon, right? And
yeah, we've been a bit up in the air with the dates of late for various reasons, and we are coming a little bit earlier this month for the next one because we're going to be in Berlin. So that's sort of we're at the Sens and Forever Healthy conference, Undoing Aging 2018. I will be there. Oliver, I hope you're there.
I'll be there. Yeah.
So you did remember to book your flights, and
yeah, I don't have my hotel finalized, so I might be slumming it on the streets, but that's okay. I will be there.
And and Keith will be there, Elena will be there, and Paul Spiegel will be there. So it's a full leaf complement, and we'll be bringing you some exciting stuff from the event. And lots after the event. And what else have we got coming up? Just too much stuff. There's far too much stuff. We're far too busy. I wish I was less busy sometimes, but then I suppose life would be boring, wouldn't it?
Yeah.
Yeah. So that's great. Well, anyway, thanks for everybody for joining us today, and for the heroes, the Lifespan heroes, for making this possible. Without your help, we couldn't do it. And yeah, we really appreciate that. And hopefully everybody enjoyed it. And we will see you next time. And thanks to Sven and Oliver as usual. And thank you everybody else.
All right.
Yep. So take care, and we'll see you next time.